Good afternoon, everybody. Thank you for joining my session. My name is Tahirah Johnson. I go to University of Maryland Eastern Shore. I study food and agricultural sciences there. And I'll be talking about the prevalence of a bacteria called Shewanella. It's an emerging bacteria that affects oysters or seafood within the Chesapeake Bay. So the eastern oyster is a key species within the Chesapeake Bay. They not only provide a outlook on the environmental conditions of the bay, but they're pretty good at providing industry work for aquaculture. So we did a study within the Chesapeake Bay, and the eastern oyster is predominantly found within the Chesapeake Bay. The Chesapeake Bay runs from Maryland through Virginia and is one of the largest estuaries in the North American region. So the eastern oysters are commercially important, and they provide a lot of insight on the conditions of the environments, of their inhabiting environments. So you're probably wondering, what is Chewinella? So Chewinella is a bacteria that can possibly cause human disease or illness. So they're gram-negative rods that are typically oxidase positive is what we looked for when we were doing our research. And they have 4 species that are pathogenic towards humans. So we have Chewinella putrefaciens, Chewinella algae, Chewinella haliotis, and Chewinella zygomatosis. And they can be transmitted through contaminated seawater or through consumption of raw or undercooked seafood. So there was one study that investigated the prevalence of Chewinella in the Delaware Bay. And what they found was that 12% of the colonies that they picked were identified to be Chewinella putrefaciens, which, as discussed previously, is considered to be a human pathogenic species. However, they used an API system, which only has Chewinella putrefaciens listed in the database, so they considered it presumptive Chewinella putrefaciens. There have been clinical samples from other countries, more so Korea, where they have isolated Chewinella, but it was often misdiagnosed as being Vibrio, which is another bacteria that is harmful towards humans. And there's limited information currently on the prevalence of Chewinella within the Chesapeake Bay, and there's no studies that have been done associating the physiochemical parameters of the water with the association of how much Chewinella is being found. So the overall goal of this project was to investigate the prevalence of potentially pathogenic Chewinella species in the Chesapeake Bay. And we also wanted to evaluate the distribution of Chewinella species in oyster and seawater, as well as correlate the Chewinella level with those physiochemical parameters. Yeah. So oysters and seawater samples were collected from June 2019 through December of 2020. We chose 3 sites within the Chesapeake Bay. We only studied within the Maryland area of the Chesapeake Bay. So from north to south, the salinity of the Chesapeake Bay increases. So Horn Point is the furthest, and we'll refer to that as HP. You'll see in the slides. Hunger River, which would be referred to as HR, and Tangier Sound, which we'll refer to as TA. So isolation of Chewinella, we use a physiochemical parameter. We use YSI to collect the physiochemical parameters, and we harvested 12 oysters from each sampling and divided those into 3 subsamples. And the subsamples, we had shoved 4 oysters into those subsamples and blended it, and weighed 25 grams of that blended meat into 225 milliliters of 0.1% peptone water to homogenize it, so we could do serial dilutions. which we then spread plated onto iron auger and incubated at 37 degrees Celsius for 48 hours. And then we'll get some black colonies you see on the screen. That will basically be presumptive Shewanella. So we pick those colonies and we will streak them onto tryptic soy auger to do an oxidase test because like I said, Shewanella is generally oxidase positive. So if it was positive in the test, we would then analyze the hemolytic activity on 5% sheep's blood auger. And then we sent presumptive isolates to GWIS for 16S sequencing. And we did statistical analysis using experiments correlation to analyze the level of if there was a correlation with CFU and the samples. And then we did Crucible-Wallace H test, as well as a Mann-Whitney U test. So a total of 802 isolates were collected, and 84% of those isolates were confirmed to be Shewanella species. The three of the pathogenic species were found, but fortunately only one of those species were in the top 4 that was isolated. So this is a graph showing, well, a chart showing the average level of Shewanella in oyster and the average in water. Now, the dark blue is showing the samples that were collected from 2019, where the light blue is from 2020. And you see that we actually had less Shewanella species being harvested in 2020 than in 2019. Now, this could be due to 2 factors. So on the far right, I made an average temperature from 2019 and 2020, and you can see that in 2019, the temperatures were higher than in 2020. But also in 2020, we had the COVID lockdown. So that means there's less water activity going on. So I'm pretty sure the turbidity levels of the water were lower during those months as well. So these are just showing the average surface water physiochemical parameters. Salinity, temperature and dissolved oxygen are highlighted because when doing our statistical analysis, it showed that they had a moderate significant correlation to the level of Shewanella species that were being found. So this is just showing the R value, P value correlation. But I wanted to point out that the water for salinity and turbidity, they had, if it was rounded up just a little bit more to the 10th, it would be at 0.5, and that would then be considered a moderate correlation, a moderate significant correlation. Now, with dissolved oxygen, it has a negative relationship, whereas the temperature has a positive relationship. So 27% of our samples collected had 1 or more betahemolytic activity or betahemolytic isolates. So if you're not familiar with hemolysin, then beta-hemolysin is usually associated with a higher chance of human pathogenicity, as well as a higher antibiotic resistance. So 33% of isolates produce a beta hemolysin and 63% produce the alpha hemolysin. And you can see that Tangier frequently isolate more alpha than beta. And I think that might be due to the salinity levels within Tangier. So in conclusion, it's safe to say that Shewanella species are widely distributed within the Chesapeake Bay. There's over 70 species of Shewanella, and we found about 10. So the top 4 species that we identified was Shewanella amazonensis, Shewanella marisfravi, Shewanella lohica, and Shewanella algae. So Shewanella algae is one of the species that is human pathogenic, but it was less than 1% of our isolates that we found to be Shewanella algae. So 37% of all isolates were beta hemolytic, and we recommend to continue to monitor the level of pathogenic growth within the Chesapeake Bay. And since Chewinella is an emerging pathogen of concern, antibiotic susceptibility testing should be investigated. September showed a significant decline in the Shewanella level for Hunger River, whereas Horn Point and Tangier didn't show a decline until October. In the study that was conducted within the Delaware Bays, they found their highest Shewanella counts being found in August. So the level of Shewanella showed a moderate positive correlation with temperature and a moderate negative correlation with dissolved oxygen. And the results from this study can help with contributing a general understanding of pathogenic Shewanella distribution, not only in the Chesapeake Bay, but in different bodies of water as well. So further studies should be done to analyze the impact of physiochemical parameters on the prevalence of Shewanella growth, because from my understanding, this is the first study to actually associate those physiochemical parameters with the growth levels of Shewanella. These are some of the literature that I used. And I just want to thank my co-authors and some of the research assistants, as well as the oyster farms that I collected oysters and seawater samples from, as well as my funding agencies. Thank you.
Thank you so much, Tahirah. Do we have any questions from the audience or online?
Hi, so my question would be, is there other types of mollusks or crustaceans that should be monitored for Shigella?
I'm sorry, are there any other?
Like mollusks, like for example, mussels that would also get that pathogen, or?
So I was studying the entire oyster, well, not the shell, but I was shucking all of the oyster meat as well as the oyster liquor, and I would blend that in the homogenate. So that's where I was getting all of my Shewanella from. But since oysters are filter feeders, I'm sure there's other ways that there can harvest Shewanella species as well.
Yeah.
Yes.
So my question is on the statistical analysis you did.
Yes.
I was wondering why rank correlation instead of normal Pearson correlation.
So we actually didn't meet the what is it called? We didn't meet the standards. Like we violated something was violated where we had to use the other test in place because our data didn't meet the correct standards for that test.
So you need to rank.
Yes.
So I love your research. Very good presentation. So these oysters, I'm not from this region, but are these oysters commonly consumed by the population?
Yeah, so Eastern oyster are the most commercially valued oyster that is being sold for consumption within the population. And a lot of people do like to consume it raw. So that's why we found a need. But not only is Shewanella in these oysters, but if somebody is being diagnosed, because we haven't found any, there's no studies to suggest that Shewanella has infected anybody within this region from oyster consumption, but due to the fact that in Korea, Shewanella is mimicking Vibrio and is being misdiagnosed as Vibrio, that leads us to believe that this should be further investigated.
Thank you.
Question. I'm not sure if you have already gone over it. Is there a response from oysters as a result of being infected by Shewanella?
I don't know. I haven't investigated that. From my understanding, Shewanella is naturally found within the environment. So that's how I feel that there's being affected. But there are some strands of some species of Shewanella that do negatively affect other seafood like fish. But I haven't dived deep into those studies since my concentration has just been working with oysters.
Okay, thank you.
All right, well, thank you very much, and thank you to all the speakers today. Really apologize for the technical issues